So my name is Marie. Nice to meet you all. I will be your host for today. And these are the two talks of our speakers. I'm going to introduce more later. And after the two talks, we will have our Q&A in the end. Without further ado, I'm just going to start now with the introduction. So, all right. So about muscle-based systems, so our motto is every cell has a story to tell. And for today, I will talk about how our technology is unleashing new possibilities in 2D and 3D in vitro electrophysiology. So for all of you who do not know about muscle-based systems, so we are based here in Switzerland, and we are offering high-density microelectrode arrays that can be used for many different types of samples. So every cell has a story to tell, and first and foremost, I'd like to mention that we have a lot of stories of our users, including, of course, János and Viva over here, who are really doing a lot of great and breakthroughs in science with our technology. So if you would like to look at the full list, please take this, scan this, and take a look at the publications. So our systems can be used in, for example, 2D culture, such as iPSC-derived neurons and primary neurons, 3D, such as organoids and spheroids, as well as brain slices and retina. And what we have observed in recent, let's say, times is that there are some trends in the field. It looks like we're entering into a new era wherein functional characterization of neural networks is kind of upgrading. And one thing that we see is that there are now new models, biological models, that are being used to understand function, whether that's development of the brain or of organs, understand disease modeling, and also drug discovery, used for drug discovery. At the same time, we also see a great upcoming efforts on engineering systems. And here, let's say the complexity and connectivity of neurons is now kind of can be controlled, and there are new models that you can now use. So these are the things that we are observing, and we are also very happy to be part of this new era to be able to reveal the new unseen phenotypes that were not possible in the past. So that is done using our next generation electrophysiology system. So we have two systems, Max 1, our single well system, and Max 2, our multi-well system. And here we have 6 and 24 well plates that are compatible with the Max 2. The Max 1 is small and flexible, can be placed inside of the incubator, you can connect it with many different devices, while the Max 2 is more for higher throughput experiments, where you have already a small chamber that is a small incubator that can be placed on a desk in your lab. So, and the core of this technology is the kind of the chips that are embedded in each of the wells. And here we have a very rich information that can be extracted by this 2 by 4 millimeter square recording area consisting of 26,400 electrodes. And it's really ultra high density in a way that there are more than 3,000 electrodes per millimeter square, and the distance between two electrodes is 17.5 micrometers. Much more important is that each of these electrodes can be connected to low noise readouts, and we can also use all of them for flexible electrical stimulation. So just very briefly about the technology. Of course, part of this is also the software, which you can use for different applications. So you can do kind of a scan of activity to know the activity of the cells and also their position. Also detect network activity and network dynamics. You can also take a look at the axonal information, meaning the action potential propagating along the axons, which can be useful to extract axonal function and morphology, and as well as use stimulation to be able to identify network excitability. So of course, these are available in our software called MASAB Live, but you can also access the data and be able to analyze it with whatever means you would want. And the steps to ensure experimental success is as follows. Of course, to functionally characterize your cells in a good way, to obtain reproducible results, to run intuitive assays, and to go beyond the state of the art. And maybe I'd like to introduce to you something that is new for us, which is our Max 1 Plus. This is our newest excellence in electrotechnology. We kind of went beyond what is already what we have in terms of a great signal quality. It's now available to order, and it's basically powered by kind of shifting, or let's say coating our electrodes aside from just platinum to have P dot. So this is a very soft material that of course is very biocompatible, has high electrical conductivity, has softness that allows the cells to really attach well, and also has long-term stability. So all of these, we have same electrode density, but now maximize performance and user experience. So before in our Max 1, we have already good user experience, signal quality, best in class, electrical stimulation, single cell level, and good surface optimization, also helped by our protocols and publications. But now this is even made better by 50% increase in signal quality because of this new surface, new PDOT, even better or more effective electrical stimulation that you can use lower voltages and even becoming more precise in your stimulation. Better cell culture viability with this new electrode material, which is actually covering 43% of the HDMA surface. So that's really highly valuable. And finally, also with sterile chips upon delivery and also automatic chip readout. So if you're interested, you can come and contact us. So in general, this is really a new chip dedicated to an improved user experience. And what's even more is that now the surface of this is something that is really flat. Of course, you can get it with either just the platinum or with the PDOT, which really allows us to have, we can now design your own networks on the Max 1 Plus. You can use structures like PDMS or other MPS devices to be able to design your own networks. I think this is also what is used by our users today in their presentations. All right, so that's it about our introduction. If you have any questions, we are happy to receive it in the Q&A. Okay, so next about our invited speakers. So let me introduce one by one. So this webinar will cover many different aspects. So just in summary, we will see talk from Professor János Vörös from the Institute of Biomedical Engineering at ETH Zurich in Switzerland. And his talk is going to be about single cell level control of neural networks on HD-MEAs for fundamental neuroscience and disease modeling. So just briefly about Professor Vörös. So he's currently a professor at ETH Zurich, and he's heading the lab for laboratory for biosensors and bioelectronics since 2016. If you'd like to know more about him, please go to our page and you can see more information. The interest of Professor Vörös and the lab is more on areas of biosensors, bioelectronics, and bottom-up neuroscience. His research group focuses on the development of novel biosensor techniques for diagnostics and drug discovery and on interacting with well-defined neural networks for experimentally guided computational neuroscience. And our second speaker for today is Dr. Viva Visalauskaite, and she's also a part of the LBB from ETH Zurich, and she studied physics in the University of Glasgow. She joined the lab back in 2024, and she's currently developing experimentally relevant computational models of in vitro neural networks for disease modeling and basic neuroscience applications. So I'd like to welcome both of our speakers and wish you a very nice webinar. Looking forward to it.
So good afternoon, evening, night, or morning also from me. Hello, everybody. The building that you see in here, this is ETH Zurich, where Einstein took his classes. But let me maybe start with another famous physicist, and this is Richard Feynman. And when he died, they made a photo of his blackboard. And what you see here on his blackboard is one statement saying, "What I cannot create, I do not understand." So basically, what he meant by this, by my opinion, is that in science we need to have some kind of an experimental system that faithfully represents nature, and then we should do some kind of measurement which provide us with data which you can use to infer a computational model. And understanding means that I should be able to predict something, and I should be able to create a model that is able to predict behavior. And if I have that and I have this experimental system, I can then start verifying my predictions with experiment. And this is this closed loop cycle that I think is also true for neuroscience. So when we want to understand the brain, this is a complex system. It has close to 100 billion neurons. And yes, if you study this human brain, that evolution optimized over millions of years, you are studying the real thing. But it's extremely difficult because you have no tools to study it properly. We have a very low resolution on humans. MRI has a cubic millimeter at best resolution. We cannot just start removing and adding cells, so experimentally we are limited. Even with the best systems that Neuralink or whoever develops, you can only measure 1,000 of these billions of neurons, which is only a fraction that you can measure. And even if you spend $1 billion that we recently actually spent in the European Union, you cannot create a proper model of this because it's too complex to model. So we now argue that it would be much better to start with something smaller, maybe 1 to 10,000 cells. And yes, the disadvantage is that this will be an artificial neural network, but we have a lot of advantages. This could be well defined. We have many intervention possibilities, and we can measure each neuron. As Mari said, every neuron has a story to tell. We actually can measure, in this case, each neuron in the network, which makes it directly comparable to computational models. And this is what we try to demonstrate today for you. So I will discuss how we do this, and then Vaiva will follow and then show you how the computational models are put up and how we use the data to actually do just what I explained here. So what is the system that we use? We use PDMS microstructures. This is a silicon rubber, a biocompatible silicon rubber. And here is the cross section. So we have these open wells where we can put the neurons. And then we have these thin, shallow channels between these open wells, which are only 4 micron, which means that the nucleus doesn't fit. So the cell bodies are not able to migrate there. This makes the system very stable. And the axons can still go and then connect to any neuron that they find on this side. This is the top view. So here you see the blue zone is the landing zone where we put the neurons in, and then their axons can grow. And then it has this special shape because axons don't like to make sharp turns. So with this special shape, we are actually primarily guiding the axon growth towards the left in this case. And it works super well. So here you see some green neurons and red neurons. And all the red neurons grow towards the green neurons, but you don't see any green axons coming back. We actually get over 90% control over our connectivity. And because it's a PDMS microstructure and Maxwell has these beautiful flat chips, we can actually put these microstructures on the chip. So here you see this 4-node network placed on the chip. And this is a video of maybe 10 milliseconds about the voltage spikes, the activity that is happening on the chip. And I would like to point out that here we don't do any averaging. So this is an added benefit of these microstructures, that in these narrow tunnels, the field is confined, so the signal to noise is amazingly good. And you can really see every spike. Now, of course, you also have the possibility to stimulate the network at any given electrode. So let's now see what happens if we stimulate this particular neural network at a selected electrode 6 times. So what you will see is that the 6 times you will get more or less the same response. Because the electrodes are so small, you can actually induce very similar responses. Not identical. These are all individual events that I'm showing, but it's actually quite nice. And why is it useful? Because it allows us to investigate the network always from a same starting position. So we don't only measure the spontaneous activity, but we can hit the network at a selected location and then gather the data and then repeat the experiment and do it again. So it's easier to explain with 4 colors. So let's say we have now these 4 color electrodes in these locations. And let's say I stimulate with the red electrode at time 0. And then afterwards, I collect the data with all the other electrodes. So green is here, and then you see that sometimes there is a spike there, black is there, blue is there, red is there. And this we can do over 250 milliseconds. And then we again stimulate the same selected electrode. It could be a combination of an electrode or just a next electrode. We register these times when the spike is coming, and then we repeat the stimulation, and then again register the times, and then we repeat the stimulation and register the time. So note that here on this axis, we have the duration between two stimuli in milliseconds, so hundreds of milliseconds. But on this axis, we can have hours and days. We can continuously stimulate and record what we see. Now, as soon as we have the Maxwell system, then under such a microarray, we have lots of electrodes that we can record from. So we have to use a lot of colors. The whole thing becomes very nice rainbow colors. And in this case, we stimulate it here. And this is then recorded for 100 seconds, the response. And you see these very stable bands appearing, showing that the response is quite stable. Now, what happens if you stimulate at different locations? So if you now stimulate at these 3 different locations, you will see that the response pattern will be rather different. So even though this is all in the same node. So something is strange. So why am I not getting the same response when I stimulate the same neuron or the same node? And the answer to this is that even though I have this implied network architecture with the 4 node on the system, this is still a fairly complex setup. It's a zoo. You have a lot of neurons, they interconnect with each other. If you zoom onto this part, even though the electrode is tiny, but you have lots of axons running over the electrode, so you never know which one is exactly that you are stimulating, or you always stimulate more than one. So now we thought about this. Okay, how could we then further improve the connectivity pattern? How could we control the intranode and intranode synaptic connectivity? So this is maybe just a staining to show that there are many synapses already within the node and there are multiple cells. Now, one possibility to do this by using the size difference between an axon and a dendrite. So while an axon is often in the micron range, the thickness is often over a micron, a dendrite is typically in the hundreds of nanometer width. Which means that if I would have now a microstructure that restricts the size to such extent that the axon doesn't fit, then I could assure to have only dendrites connecting to the axon from one location of the chip to another. So here we have 100 nanometer order holes between 2 compartments where the dendritic spines can go across and connect to the axon, but the axon doesn't fit. And here is then the realization. We often get the axons into these long channels to assure good connectivity between these setups. In a larger view, you see that the dendritic compartment in this case has such a shape, and then the incoming axon is wrapping around it. And between those zones, you have these nanochannels that are not visible on this microscope image. If you zoom in, you might see some of those connections between. Now, whether these are functional or not, you can check by calcium imaging. So what you see here is a video, and here is the incoming axon. And when it's active, you see that sometimes also the dendrite on the other side becomes active. It's not so easy to see in the video, but if I zoom onto different regions of interest, so then I can show that here is the blue is the axon region. This is the activity that we observed in the calcium imaging. Here is the spine head. You see that not every spike, but most of the spikes are represented. The spine neck, already some spikes are missed, but many are there. And then finally the dendrite, you will pick up some of those spikes, showing that you can have the information transferred between these. Now, this allows you to start building very simple networks. So you could have networks between 2 neurons, so 1 in and 1 out, or between 3 neurons that you have 2 input neuron and 1 output neuron, and you can then put these synapse formation zones at the location where you want. And just to show you that it's actually possible, here you see a Max1 chip where you have 200 these 2 neuron networks. And if you zoom on this, then you really see that they are nicely ordered. And on this video, you actually see how the action potential propagates between these 2 neurons and then also goes across. And this provides you with a lot of possibilities. For example, you could start now investigating the latency between the presynaptic and the postsynaptic neuron. So whenever the presynaptic neuron fires, we put it at 0, and then this band is the postsynaptic neuron. And then apply some kind of stimulation protocol and see what happens. In this case, the latency did not change, but the synaptic probability, the transfer probability improved dramatically. And that was even staying for a while. So it's a system that allows us to systematically study synapses between two neurons. And you can also then test all the standard textbook potentiation protocols, and you can then choose your neuron type. So it's really a very nice system that allows us to collect a lot of data. So this is very useful for fundamental neuroscience. But the whole concept can also be used for modeling diseases. And one of the diseases that we try to model is a nasty brain cancer, glioblastoma, that is affecting neuronal networks over time. For this, we again use these polydimethyl siloxane microstructures. Essentially, this is then the architecture. What we do, we have in this case 2 compartments where we can put the neurons, and then a third compartment where we can put the primary tumor from patients. Now, the axons of these neurons are forced close to the microelectrode array by these axon channels. And there you can do all your recording with very good signal to noise. And the axons also reach this compartment before we add the tumor. And when we add the tumor starts to interact with these axons. Here we have a little video about this, how the tumor traced with the red dye is interacting with the axon ends that are traced with green. And as you see the video, the cells are really approaching the axons, and the whole axon ends are then engulfed. And even more, because it's a tumor, these cells are able to soften the nucleus and even enter the channels and start interacting with these incoming axons in these microchannels. Now, the whole thing is very robust and high throughput in a sense that we can put multiple of these systems onto the chip. So here you see many of these architectures on the chip. And you can start recording the data. So here you see just an example for the video that we recorded. Often we actually analyze the data very carefully. So every spike matters, not just every cell. And for example, in this glioblastoma, we notice that if you just have astrocytes, not cancer cells, as a control on this compartment, then the activity pattern stays stable over many days. So this is diff 37, this is diff 47. There is no much change. But if you add glioblastoma, then the standard activity pattern becomes much more bursty, and also these burst firing rates are huge. So there are many differences that we can observe with these added glioblastoma, and we even see patient-to-patient differences. So in summary of my part, I hope that I was able to show you that this bottom-up neuroscience concept is very useful as a tool to do fundamental neuroscience, but at the same time, we can also apply it for as disease models. Why is it useful? Because we can have stimulation and recording, and we can stimulate any neuron and we can record any neuron or every neuron. We have an extreme control over the local environment. We can have very stable recordings over days and weeks without any problem. We can add drugs. We have some papers, if you're interested, you can look it up. And you can also produce the highly reproducible data to generate computational models that Vaiva will show you. And these models can then be verified with further experiments. Because what I showed is all human neurons, so these are patient-derived neurons. This has also a 3R potential and a better translatability, hopefully, towards true medicine. And finally, I had no time to talk about this, but it also allows us to start connecting computational neural networks to biological neural networks towards hybrid intelligence. So before finishing, I would like to acknowledge the funding sources and the team that is behind the work. In this particular work, majority was done by Katarine and then Julia. Let me find her here. So thanks a lot for your attention.
So I will continue discussing in vitro platforms, but maybe first of all, hello everyone. It's nice to see so many people. My name is Vaiva once again, and I will continue discussing how we can use the data collected from these in vitro platforms to develop in silico modeling approaches, specifically concentrating on models that are related to synaptic transmission in healthy and diseased circuits. Why is it important to understand synaptic communication? Well, for one, it's basically at the heart of everything that the brain does, from perception to memory to movement and emotion. And when this communication breaks down, the consequences can be dire. Disorders of the nervous system as diverse as Alzheimer's, autism, epilepsy, as well as diseases associated with support cells of the nervous system involve possible disruptions at the synapse. So to better understand and ultimately treat these conditions, we need tools that let us observe, control, and model synaptic function in a biological experiment. To translate these experimental models into treatment strategies, we need biophysical models grounded in these experiments. And this combined in silico and potentially in vitro framework allows us to reveal mechanisms driving observed activity changes. And while our ultimate goal is to understand the brain is arguably the most complex system known to man. And instead of trying to understand the whole system of trillions of neurons and synapses, in the lab, we use the so-called bottom-up neuroscience approach, which considers simplified in vitro systems. Here, once again, we use patterned in vitro networks of human-induced pluripotent stem cell-derived neurons cultured on high-density microelectrode arrays. So connectivity between neuron populations is controlled using the PDMS microstructures, and this allows us to develop networks of interest, networks that are tailored to isolate specific mechanisms of interest. So altogether, we have a platform which allows us to have highly reproducible data with human relevance, and importantly, through the use of PDMS microstructures, we can isolate the neurons and record from them at the high spatiotemporal scale. Resolution. So with this experimental framework at hand, let me try to address the first question that I'm asking in this presentation. It's a very broad question, but we'll try to shed some light on it. How does the brain process information at the level of synapses? So to precisely study synaptic transmission, in the lab, we isolate just a single presynaptic and postsynaptic neuronal pair using the PDMS microstructures. Each microchannel hosts a pair, and we can record for many of such pairs in parallel using high-density MEAs. The synaptic transmission between the presynaptic and the postsynaptic neuron can be confirmed almost immediately visually, but also pharmacologically and using statistical tools. I'm also asking in here, can we also explain what we see in the experiment using a biophysical model? So the goal of this biophysical model is to emulate the experimental setup as well as possible. And specifically to do so, since our cells are constrained within microchannels, it is sufficient to model them as simple so-called ball and stick multi-compartmental model of neurons consisting of a single soma, so of two compartments, one for soma and one for axon. And since we want to model traveling action potentials, we need to have multiple segments in both the soma and an axon, and each segment includes active and passive ion channels that allow the action potentials to propagate. And we use model parameters that are relevant for our human-induced pluripotent stem cell-derived excitatory human neurons. Very important for this model is a definition of a synapse, and here we model synapse using a conductance-based model with two components, the fast AMPA and slow NMDA components. Importantly, this synapse is located somewhere somewhere along the axon of the postsynaptic neuron, and when the traveling action potential from the presynaptic neuron reaches the location of the synapse, sometimes it triggers the synaptic event at the synapse. Okay, and lastly, to elicit spontaneous activity, we administer a pulse-on input at the axon initial segment at both neurons. So now this is how the model is set up. The next thing is to fit the model to the data. And formally, what we're trying to do is to perform statistical inference, whose goal is to find the so-called posterior distribution. That is the most likely parameter values of the model that produce the observed data. Once again, the model parameters include all the variables within the model, such as diameters of the axon and the location of the synapse, the strength of the synapse, and so on and so forth. Now, the raw data is the MEA recordings. But what we really want to understand is some key statistics that we can derive from this data, and we're trying to find the models that are consistent with these statistics. So specifically, the statistics that I'm talking about is first two pieces of statistics related to the properties of the neuron itself. So first, the firing rate of the neuron and the conduction speed, which we can measure experimentally as well. And the last two are related to the properties of the synapse. So the first one is the coupling lag. So if we observe the spike at the first neuron, so the presynaptic neuron, with what lag do we also observe this? Do we observe spike at the postsynaptic neuron? And the fourth one is the synaptic probability, which quantifies how often does the activity at the presynaptic neuron elicits activity at the postsynaptic neuron. So since we have a computational model, inferring the model parameters, so inferring the posterior distribution is actually very difficult. And one possible way to do it in a simple way, in an efficient way, is using the so-called simulation-based inference, which uses forward simulations to approximate the posterior. So the way that it works is first, we need to define a prior distribution of model parameters. So all the biophysically plausible ranges for all of the parameters that we're willing to test. And then we need to run forward simulations using parameters sampled from this prior distribution. And again, in the simulation, we can generate measurements as we would do in the experiment, from which we can derive the summary statistics that we're interested in. And using this library of model parameter and summary statistic pairs, we can train a neural network which approximates the posterior distribution. So after training, model parameters can now be inferred for all of the biological synaptic pairs. And how do we do this? So for each of the biological synaptic pairs, we again calculate some summary statistics of the real data. And then we sample from this posterior distribution approximating neural network to infer the most likely parameter values that are consistent with the data that we see in the lab. So let me first demonstrate that the inferred models actually reproduce the dynamics observed in the experimental data. And here you see one so-called spike type triggered raster plot, which essentially shows you that if we record the activity at a trigger electrode, here the one that is close to the presynaptic neuron's soma, and we align all of the spikes recorded at a downstream electrode close to the postsynaptic neuron soma, we see that the spikes happen with persistently, so we see that there are two latency bands appearing. That is that whenever a presynaptic neuron spikes, we see some spiking activity with a delay of 2 milliseconds and 4 milliseconds close to the postsynaptic neuron. And we hypothesize that one of those latency bands is axonal propagation of the presynaptic neuron, and the latter one is due to synaptic transmission. And we can directly validate this hypothesis using an inferred model. So the model the model that most likely produced the data that we have in the lab also has very similarly looking spike time triggered raster plots. However, unlike in the experiment, in the simulation, we can easily deduce the sources of the electric fields in each that are recorded at each electrode and confirm that the first synaptic band at 2 milliseconds is indeed axonal. So we quantify the model accuracy by computing the absolute deviation between simulated and real summary statistics across all of the pairs that we have measured from in the lab, which is over 3,000 of such pairs. And we see that the absolute deviation between real and simulated statistics is below 10%, which for the most likely parameter values. And that indicates that the model is both accurate and generalizable. So with the model validated, we also use it to probe synaptic mechanisms. And specifically in here, we studied long-term potentiation, which is a key process by which repeated coincidental activity strengthens synaptic connections. And in here, you see an experimental setup. So to elicit long-term potentiation, we applied 5 Hz simulation to both pre and post-synaptic neurons and recorded spontaneous activity before and after the simulation. So with the inference technique that I introduced, we inferred the most likely parameter values before and after the simulation. And here you see the distribution over all of the experimentally recorded data, all of the experimentally recorded pairs. And you see that there are key changes that are due to the LTP protocol. First, we see an increase in strength of the AMPA synaptic component, which is consistent with early phase LTP. Second is the decrease in the strength of NMDA component, which is also consistent with early phase LTP. And then we also see some reduction in the intra-event interval of the presynaptic neuron, which is possibly due to homeostatic response. There are also some other secondary alterations that we see. So just to finish on this, altogether, the model not only detects a potentiation, but also reveals its likely biophysical sources. So far, we've considered synaptic transmission and plasticity in healthy circuits. But what happens to synaptic transmission when these circuits are diseased? And one case that we explored specifically is related to neuron interactions with glioblastoma. And as was already presented, glioblastoma is an aggressive primary brain tumor that arises from glial cells. So there's a striking shift from the older view that glioblastoma simply displaces or destroys neural tissue. Now it's clear that the tumor and the neural circuits are very tightly connected, and glioblastoma cells also may form functional connections with neurons and synapse-like structures. So these interactions seem to form a vicious cycle where glioblastoma cells drive neuronal excitability, and in turn, the increased excitation of the neurons leads to faster glioblastoma progression. And while it's quite arguably, there's a lot that is understood about glioblastoma at the cellular as well as whole brain scales, it is still not that well known what kind of alterations does it have on the neuronal communication at the level of local brain circuits. So this is exactly what we're trying to understand in this project. How does the presence of glioblastoma alter activity and connectivity within local brain circuits? And how does this reorganization unfold over time? So once again, this is exactly the level of resolution that our in vitro platforms can target. And let me remind you about the platform that we use for this project. Specifically, we have a microstructure that is composed of 3 wells connected through two sets of microchannels. And neurons are placed within 2 of those wells. And as they mature, they grow connections between the 2 wells and into the 3rd empty well through these microchannels. And once the network is mature, we introduce primary glioblastoma cells from 3 patients into the 3rd well, and neurons start to interact with those cells as well. So already from simple data analysis, we can see that glioblastoma has striking effects on spiking activity. So if we look at the astrocyte and neuron co-culture, which is one of our controls, we see that the population firing rate, so the total number of spikes that is observed within a small time window across the whole culture, this seems to be quite regular, and the population firing rate does not seem to change between 37 and 47 days in vitro. However, if we look at the glioblastoma neural co-culture, we see these seizure-like epileptic bursts appearing. And another important thing to note in here is that there seems to be a big heterogeneity in the dynamics across the 3 different patients. One possible way to explain this is through the fact that perhaps, so one hypothesis that we have is that glioblastoma reorganizes the neuronal circuits. And one way to probe or to test this hypothesis is to use some tools from network science and information theory. Specifically, we model the network of neurons as a network where each neuron is represented as a node in the network, and then its effects on another neuron are indicated through edges. And we can infer, since we have a lot of data, we can infer the whole network of interactions between those neurons and try to understand its topology and how it's changing due to glioblastoma. So the way that we infer this network of communication between neurons is through tools from information theory. So let me give you a very short, very quick primer into information theory. At the heart of it is the concept of entropy, which quantifies the uncertainty in determining the state of each neuron. And if neuron state is never changing, for instance, it's always no spike or always always spike, then such neurons spike train has no entropy or very little entropy. But in reality, it's whereas on the other hand, if the neuron spikes completely randomly, it has a very high entropy, and we have no hopes in trying to predict anything about its activity. However, if the activity is somewhere in between, we can use other information available in our network to predict the state of a neuron at each point in time. So first source of information, it's neuron's own past, and if some k-past states of the neuron are useful in predicting its current state, so spike or no spike, we say that there's presence of active information storage. Another source of information is other neurons' activity of other neurons in the network, which we can again quantify through the so-called transfer entropy, which measures the reduction in uncertainty in accounting for the state of another neuron L times steps in the past. Also, there are higher order measures that we could use, which I will not discuss in this talk due to time constraints. So first, let's look at transfer entropy. We can measure the amount of transfer entropy in the network associated with each point in time. And here I'm showing you the sum transfer entropy across all pairs of neurons in the network, overlaid with the population firing rate. And what is important to note in here is that there seems to be elevated information transfer during the burst. So neurons tend to prefer communicating, quote unquote, with each other during these highly synchronized states in the network, in time. And here again, you see that the outside of the burst transfer entropy is a lot smaller than within the burst. Furthermore, if we compare the results between control and patient conditions, we see that the activity outside, so transfer entropy outside of the burst is significantly lower than in controls and significantly larger than in controls within the burst. What could be driving that kind of activity? One possible way to explain this would be by looking at the inferred network structure. So recall that with transfer entropy, we can calculate it for all the pairs of neurons that we have in the sample. And from this, we essentially get a whole network of interactions. And one very simple network measure is degree distribution. And in here, I'm showing you our degree distribution for every node in the network. And essentially, the out degree distribution, as I illustrate in here, shows you how many out neighbors does every neuron have. And essentially, if the neuron has a lot of these outgoing neighbors, then it means that it has a lot of influence on other neurons in the network. So out degree is the number of outgoing edges. And what we see in here, that in controls, this degree distribution is actually quite homogeneous. So, well, a lot more homogeneous in control and astrocyte conditions as opposed to patients. And the second thing that we see that is important is that degree distribution becomes more fat-tailed in patients A and B. And what it means is that there's emergence of high degree nodes, which can act as hub nodes that activate many downstream neurons and initiate these bursts. So very briefly, because I think now I'm a little bit out of time, glioblastoma also has effects not only on how neurons interact with other neurons, but also how they integrate their own past information when calculating their spiking behavior. We see that active information storage is substantially higher compared to controls. And the temporal window over which the past influences the present is also broader. One way that we could explain this is by looking at small subgraphs in the network, specifically if there's presence of a lot of feedback, if there's presence of a lot of or more emphasized feedback in the network, this could enable reverberation of activity and in turn increase memory of neurons. So as we see, the feed-forward node cycles shown on the left don't seem to have increased at all in between the control and patient conditions, whereas the feedback cycles, which should help with reverberating the activity, are significantly larger in patients A and B. And here again, the patient C is an outlier, where we see that feedback cycles did not really increase in prevalence in these networks. So the lack of these changes may also explain the absence of bursting activity in this network in particular. This suggests that glioblastoma-driven alterations in network dynamics are linked to structural reconfigurations. And of course, to validate any of those hypotheses, we'll have to come back to mechanistic modeling again. So just to sum up, first, in the first part of the talk, I showed you a mechanistic modeling approach that helped us reveal biophysical processes underlying synaptic transmission. And in the second one, I discussed how one could use information theory and network analysis to characterize alterations in network structure in glioblastoma infiltrated networks. Thank you again.
Thank you so much. Thank you, Janusz, and thank you, Viva. Really, really beautiful story you have here. So, starting from designing microchannels, and also very exciting experimental design, and then moving forward to biophysical modeling, and then even disease modeling all in one work. So really, really wonderful. Now I'd like to welcome questions. Please add them in our Q&A. So the first question that we have is from Frederick Seibd. Hello, Frederick. This is a question for you, János. Regarding data acquisition, especially analysis, did you use the muscle biosystems tools or is it all custom? If so, which effort did it take to get all the results?
So we do use many of the Maxwell available tools, but of course the one of the most important one is the interface that allows us to develop our own Python code.
Yeah.
And whenever we publish something, we immediately publish the code along. So this is not trivial to do these things, but it's also not rocket science. So it can be done. I think with time, this will become smoother and smoother.
Thank you. Thank you so much. Um, all right. Let me go to the next question. I hope that answers your question, Frederick. Next is from David Yekel. Hello, David. Fantastic talk. He said, in such small networks of two single neurons without further input, would you really expect spontaneous activity? Or he imagines that the missing input would lead to silence or no activity. Any
Maybe
of
I take it way as well. The data that we presented were all mostly spontaneous activity, and in fact, the frequency of spontaneous activity is not much different than in a more complex network. So on average, the spike frequency would be very similar. This is why we had to introduce the noise parameters, because these neurons are very active also spontaneously.
Thank you so much. So we have one question from an anonymous attendee for Viva. Regarding mechanistic models, are you planning to emulate full networks and integrate glioblastoma as well? So which kind of challenges do you envision in particular regarding high dimensionality? Thank you.
Yes, and so this is the next challenge. How could we now depart from just a pair of neurons and simulate the whole network? And as the anonymous attendee highlighted, the high dimensionality will be the biggest challenge. Perhaps we can learn something from network science, because in there one can show that, well, perhaps not even network science, but also physics, where perhaps we could simplify some of the models and still have good enough, well, explainability. So we'll try to seek for parsimonious models. And regarding integration of glioblastoma, yes, this is also the next step. So to try to explain all that we see from the data analysis with the modeling. And here, I think it's a little bit less well documented how to model the glioblastoma itself, whether it's a field or do we also model them, model cells as individual cells.
Thank you so much. And then I think the next question is for both of you. You can choose whoever will answer. Another one from an anonymous attendee. Did you investigate the change in the prevalence of other network motifs in your work with glioblastomas? And did you distinguish between excitatory and inhibitory neurons?
So maybe I can try to answer this. Yes, so we explored at the moment, not all, so you can count the number of different motifs of different size. I don't remember exactly the precise number of motifs of size 3 or 2, but you can count them and then run the same calculations for all of them. For time constraints, I didn't go through all of them, but it seems to be that a lot of feed-forward motifs are not really changed due to glioblastoma. And then feedback motifs of different sizes also seem to have increased in prevalence. And then to discuss regarding the distinguishing between excitatory and inhibitory neurons, that's very easy. We only have excitatory neurons in our cultures at the moment. It's a given.
But we do have a good source for human iPSC-derived inhibitory neurons from Amsterdam. So hopefully very soon we will also be able to show excitatory inhibitory balance.
Looking forward to what's next then. Thank you. Another question from David. Amazing talks. Thank you. He was wondering if the memory is longer and stronger in neurons with glioblastoma, and does that also make the existing network stronger?
So that's probably a question for me. And to be honest, at the moment, I don't know. I would need to check. We're still in the process of analyzing all of the data.
Thank you so much, Vaiva. We have more and you're very popular talk. So another question here. So from Kioma Okimbata. So hello, Vaiva and Janusz from Kioma. So thanks for the presentations. The question is from the diagram of information theory and network analysis. What is the implication of excitation and very reduced or no excitation to the neurons and glioblastoma?
I'm not sure that I understand the question. Would it be possible to
Kioma, would you like to speak? We can allow you to speak if you're okay, or you can type.
maybe now I understand. So is it
Okay.
happens when the neurons are very excited or there's a low excitation? Yep, it would be nice to clarify
Yeah.
it.
Well, let us know how best you'd like to follow up. Either we have you talking or you can write us a comment. We'll move on to the next question from now. So the next one is from Mutiyat Usman. Hello. Would like to ask about how you handle missingness in your research and handling of outliers from the box plot and distribution plots you showed.
Um, you wanna shall I because so basically we show all the data. This is why we have these box plots.
Yeah.
And because we have millions of spikes that we analyze, and also this is why the distributions are so broad. We can still do a significant test and deduce serious conclusions from the data. So this is exactly why we need lots of data because biology is inherently heterogeneous and the distributions are inherently broad. I never dare to start computational neuroscience models before knowing what is actually present in nature. Now that we know these distributions, it's easier to set up realistic models.
Thank you. Yes, I hope that answers the question, Mutiyat. Next, we have a question from Jose Mateus. But he mentioned that it's already replied, so no worries. Next is from Bin Lin. For the LTP study, what do you mean strength, amplitude, frequency, or area?
So in the model, we can vary the strength of the contribution of the AMPA, so fast or slow NMDA component of the synapse. So it's really the strength in the model, or in other words, the weight.
Thanks a lot. And we have one last question right now. So from Tao Huang. Hello, Tao. So asking, have you used AMPA or NMDA blockers to dissect the contribution of each type of receptors?
So I think we used just general synaptic blockers that completely blocked the synaptic transmission altogether.
But of course, I mean it's straightforward to do, and it's on the to-do list to block specific receptors.
Thank you so much. Thank you so much, everyone, for participating in this Q&A and also the full webinar. So just a few announcements. What's next? So we have another webinar together with Fujifilm Cellular Dynamics on July 16th. So more details to come. Please wait for that. Also, we are our postcards are traveling around the world. If you have them and you're visiting a place, please send us a copy and or post in social media and tag us. That would be super cool. Also, we're going to be in Vienna next month. So I think Professor János Vörös is also going to be a speaker there. And we have our Maxwell Symposium, aka our user meeting. These are the confirmed speakers. We still can have some registrations. So if you're interested and if you're right in the area, or if you would like to travel in Vienna and join us, please scan this QR code to learn more, or go to our website and you can see it at the lower right corner on the home page. Also, after our user meeting, we are going to have the MEA meeting, also in Vienna. So it's just in the same location, so you can combine your trip into one. And finally, just to let you know, if you find yourself in Japan, so we have actually an exhibit there together with University of Tokyo, wherein they integrated multiple of the organoids on top of our arrays. And we're also going to have an open event on August 4. So thank you so much, everyone, and looking forward to see you in the next webinar. Have a nice evening or day or afternoon, wherever you are. Thank you so much, Janusz, and thank you so much, Vaiva.